We'll be talking about cell communication. Cell communication is when cells send messages to one another through signaling, usually by chemical means. The main type of communication that we're going to talk about is the signal transduction pathway. Now, this has three parts to it. You have the receptor, which is the first part. Then you have the transduction, which is your middle part. And then you have the response. So the first part, like I said, is the receptor, and this is when a molecule is binding itself to the receptor on the plasma membrane. So this is what is connecting, and then you have transduction. That molecule that is connecting, it has a message, and what transduction does is it translates the message so the cell can understand what it is being told or asked or whatever is trying to be communicated. And then you have the response. And then the response is the cell actually responding to whatever the message was translated in transduction. Then you have the ligand. And the ligand is the actual molecule that is being bound to the cell membrane and is being translated and is creating the cellular response or the chemical reaction in the cell. So every time that a molecule is being accepted into the receptor is going to be a ligand. Now I'm going to talk about the receptor protein pathways. And you have two main ones. And the first one I'm going to talk about is the G protein receptor pathway. And what this is the G protein is an enzyme or protein that carries a cell message to where it needs to go, to a certain place. And so you're going to have a couple parts of this, and the first one is going to be the G protein linked receptor. And this is where the signaling molecule attaches to the receptor in the plasma membrane on the cell. So what I'm going to do is, we're going to speed up the video, and I'm going to draw the cell membrane so it makes a little bit more sense. So this is, you have your plasma membrane and then your cytoplasm right here. So the first thing that's going to happen in the pathway is that you're going to have a signaling molecule, which is your ligand, and you're going to have that just come right in the receptor protein. And it does this because it can't just enter the cell. It has to go through the receptor. And so what it's going to do is we're going to put ligand up here. And once this enters the G protein receptor, this G protein down here with the GDP, this is inactive right now. So when this ligand, the signaling molecule, comes into the G protein receptor, it's going to take that G protein and it's going to go through phosphorylation and it's going to travel up and go right into the receptor. And so you're going to have your G protein in here. And it's going to drop this ADP, so this little guy's going to come down here. And what it's going to do is it's going to attract or make a GDP. And GDP is very similar to ATP, which is just energy. And so this causes a shape change in the G protein receptor. And so what's going to happen now is we have our G protein, and it's going to come right along across the membrane right here to the enzyme. And what's happening now is that the enzyme is, once the ligand enters, that's the receptor, the first part of the signal transduction pathway. Then you have the G protein becoming active, because it's active up here. And this is transduction, because the cell understands what message is being carried, and so it's going to travel across to the enzyme where your cellular response is going to take place, if there is a cellular response. So this is a recurring process because it is repeated. Once it gets to the enzyme, what it's going to do is it's going to hydrolyze, and basically, let's just erase all this. Basically, it's going to take that active G protein, and let's say if it's down here and it goes through that shape change, G pro, then it's going to it goes through phosphorylation, so you have your P, and then it loses that ATP, and so what's going to happen is it's going to go back to an inactive G protein with the ADP again until it gets another signaling molecule. And this is kind of how the G protein receptor pathway works. So now what I'm going to talk about is secondary messengers. And so what that is your ligand, that's your first messenger, right? You have it entering the receptor, and its transduction takes place, and you're understanding what the message is, which is then being sent over to the enzyme to fulfill that cellular response. And so then what's going to happen is if you go further down, so we have the GTP and the G protein, we're traveling across the enzyme. Now what we're going to have happen is the G protein is going to activate a conversion, and what it's going to do is you're going to come down here and it's going to make, let's see, you're going to have ATP, which is just like the A, or the GTP, and it's going to do a acyclic AMP, which this right here is going to be your second messenger. What we're just going to do. This right here is going to be your second messenger, and this is because what it's going to do is this right here, this is going to go down and activate another enzyme in the cell, and it's going to create more cellular responses. And so that is why it is the secondary message, because ligand right here is going to be your first message, and then once you go through the first enzymatic process, then you're coming down here to start another enzymatic process, which usually you can use the protein, let's see, kinase, and then that's going to go to more cellular responses. So the second pathway that we're going to talk about is tyrosine kinase pathway. Let's see, tyrosine kinase pathway is involved with repair and growth in cells. This process can activate many relay proteins at once, and this is because each one of these little tyrosines right here, these are each its own separate unit, and it's its own separate monomer. And so let's say that this time the ligand, we're going to say if it's a growth factor, let me just write growth factor right here. If it's a growth factor, you have your inactive receptors. And so what's going to happen is that when these growth factors come in, they're going to turn partially active. And what's going to happen is once they're partially active, the monomers, these right here, the receptors, are going to connect together or come very close. And this activates the tyrosine partially, where you add a phosphate, let's see, for each separate unit. And you get to add a phosphate from ATP, because you have 6 ATP and you have 6 separate units. And so with fully activated phosphates and tyrosine, before you get the phosphates, you have an unphosphorylated receptors, and then once the ATP gives you the phosphates, then it becomes phosphorylated, and that happens when the relay proteins recognize the tyrosine and bind with them to make multiple cellular responses. So what's going to happen is you're going to, let's say, you're going to have a couple relay proteins, and each relay protein that you have, it's going to, so if these are relay proteins, what it's going to do is that these relay proteins, they're going to have a certain one of these connect to. So this one's coming up here, then it's going to just fit real smoothly around. And so when that happens, then you have your cellular responses. Okay. Next, we're going to talk about ion channel receptors. Ion channel receptors are, they're also called ligand-gated ion channels. This is because what you have is a control of like a particular or a certain signal. See, ion channel receptors. And so these guys are going to be targeting only certain cells. and only have messages for some or need to respond to some. So what's happening with ion channel receptors is that they control, like I said, a certain signal. And what's happening is this little thing right here, it's a gate. And so when we get the, we'll use this color. When we get the ligand in here, what it's going to do is once it reacts to that, what it's going to do is it's going to open this gate. And once this gate is opened right here, and this gate is the the ligand gated ion channel receptor. And so what it's going to do, it's going to send the ions, fill this back in a little bit. And what it's going to do is going to send the ions right down through the membrane, and it's going to send them into the cell's cytoplasm. And once they come in here, what they're going to do is they're going to start depolarizing that cell. And these are, once the gates open, ions can go in, but as soon as your ligand removes, let's see if we take this out of the equation, then that gate is going to open back up. Or it's going to close back down, and we're going to have to wait for another ligand to come back through before we can open it back up for more ions to enter. And what happens is that these ions, what they're going to do is they're going to rapidly change the concentration of certain things inside that cell to get the cellular responses. The last thing we're going to learn about is intracellular receptors. These receptors are they're mainly for like steroids and for hormones, and they don't have to have the receptors in the membranes like the other pathways did. They can just diffuse right across. Let's see, receptors. They can just diffuse right across the membrane because they're not proteins, they're lipids. And so right now I'm going to draw it and speed it up a bit, and then I'll come back and explain it. Like I said, the intracellular receptors, this is for mainly hormones and steroids, and in my example, I'm going to use a hormone. And so, like I said, it doesn't need, it can just diffuse right across the cell membrane. It doesn't really need the receptor in just crossing because it's not a protein. Hormones and steroids, they're lipids, and so they can just skip that process. And so what it's going to do is you're going to have your hormone, and it's going to travel right through. And then your cell receptor is already going to be inside the cell. And so what's going to happen is it's going to come over, and it's going to have that. And you're just going to have the hormone, and it's going to be inside that receptor. And once it does that, it's going to turn into a hormone receptor complex. And this, it eventually, what it's going to do is it's going to, I'm going to keep it in purple. It's going to travel in the nucleus right here. And it's going to kind of act as like a transcription factor. It's kind of going to be able to let's see, it's like stimulating the transcription of like the DNA, and then it's into the mRNA gene. And so it's going to come down here, and then, like I said, down to the RNA. And then once that happens, it's going to be able to leave the nucleus, and a new protein is going to be made. And so if I could just draw the new protein. This is going to be the one. And this protein, it's already going to have a job set up for it. It's already going to know what it's supposed to do and how it's supposed to function. And so see, that's it, and then you're going to have the new protein. Intracellular receptors, they kind of mainly start the making of RNA, and so that, let's see, new protein. And so just go back over it. You have your receptor protein, then your hormone, and it's going to come through, and it's going to do the receptor protein hormone complex that's going to travel inside the nucleus, and it's going to transcript with the DNA right down to the RNA. when it's going to be able to make a new protein.